The topic for today is the microbiome gut-brain axis regulates social cognition and craving in young binge drinkers. This study, together with some other recent scientific reports, is beginning to deal with the effects of binge drinking on the enormously important gut microbiome. Dr. Gonshor, thank you for doing this.
Thank you, Reed. As I mentioned in our last broadcast, this particular one and the one prior to that was dealing with microbiome. We're going to be going on to a lot of stuff on glucose intolerance, insulin resistance, all kinds of other stuff in the coming weeks. So just to let you know that's coming up. But remember that in each of these sessions, we take a deep dive into recent research that brings to light objective scientific evidence on whatever the topic at hand happens to be. And so today we're looking more closely, as Reed said, at the microbiome gut-brain axis and how that regulates or helps regulate social cognition and craving in young binge drinkers. Now, this study, like other recent research, is beginning to clearly show that binge drinking in adolescence, which is a problem, has deleterious effects, not only on gut function, but on emotional processing and social cognition, very important issues. So let's look at the actual evidence because, as Reed is going to say,
Science matters. Now, after our last broadcast, which was titled Systematic Evaluation of Antimicrobial Food Preservatives on Glucose Metabolism and Gut Microbiota in Healthy Mice, there were some questions, as you might have expected. And when these come to our attention, we always like to make sure we pick out the best one or two questions and get those answered by Dr. Gonshor in the follow-up program. So one of the questions was, Dr. Gonshor, can you explain in greater detail, please, how biologic antimicrobial preservatives affect glucose metabolism?
the first thing I want to say again is, Reed, I don't know if you remember, but this was a topic you suggested. This was the actual paper that you sent to me. So if people think we're not actually talking to each other and looking into these things, we do. And in this study that we highlighted, it was shown that both synthetic and biogenic antimicrobial preservatives, these APs, induced glucose intolerance in
Hmm.
healthy mice. And remember, wild type healthy mice in many ways are very much like humans in terms of their metabolic systems. So this is important. Therefore, the chronic exposure to these preservatives alone, or as you quite often see them in combination, may be a concern, especially for the people with glucose metabolic disorders to begin with. For them, it could be a real problem. And nisin, which is one of the preservatives that we talked about, is an antimicrobial peptide, and it's produced by Lactococcus lactis, one of the commensal bacteria that one has in the microbiome. Nisin induces gut microbiota alteration, and it strongly downregulates or decreases amino acid formation. And I don't know, for those who heard it last time, we discussed it in some detail. Gut microbiota dysbiosis has been linked to many metabolic disorders, as well as impaired amino acid metabolism. And why is that important? Because if you downregulate, if you decrease amino acid metabolism, it might further change the GLP-1 release, and that has an impact on metabolism. GLP-1s, as you know, which is being used for weight control right now, it's a monster series of drugs in the world. GLP-1 has multiple metabolic effects, such as glucose-dependent stimulation of insulin secretion, suppression of pancreatic glucagon secretion,
Hmm.
a decrease of gastric emptying, and an inhibition of food intake. That's what makes the GLP-1s such a monster drug today. So decreased GLP-1, which is induced by nisin, could further impact the secretion of these glucoregulatory hormones, namely glucagon and insulin. And in that way, it can impact glucose metabolism. So remember, all of these preservatives and so on, which are ubiquitous, you may say, well, so what? Well, the so what is that it has an effect on things like amino acids, which then has an effect on GLP-1s. And then you run into the whole plethora of problems that you see both in weight control and in diabetes, which is where the GLP-1s were originally developed for. And that's it.
Thank you. And fantastic. We do a lot of gut testing in FDN, and love to see these deep dives into the science behind it. So thank you, everyone, all of you, for submitting your questions. They're very important as we learn together. And Dr. Gonshor, now let's jump into today's study about the microbiome gut-brain axis, how that regulates social cognition. Maybe you could explain that to us, and craving in young binge drinkers.
Well, let's take a look and see. Well, thank you, everyone, for being here today. And we're going to discuss, as Reed said, the microbiome gut-brain axis regulates social cognition and craving in young binge drinkers. And this is a study that came out very recently, 2023, a few months ago. And what they're saying is that the research demonstrates alterations in the gut microbiome of young binge drinkers and identifies early biomarkers of craving, associations between emotional processing and microbiome composition. You wouldn't have thought that those two are together. Supports growing literature on the gut microbiome as a regulator of social cognition. That's very interesting. So what about this binge drinking, etc.? Well, the microbiome we know is essential for proper digestion, for immune function, intestinal health. We've known that. We also know something about people with alcohol use disorders, AUD. It's defined as an inability to control or to stop problematic drinking. They often have dysbiosis, which is associated with greater inflammation and disease compared with healthier microbiomes. So these people with an alcohol use disorder are shown to have dysbiotic issues. So maybe alterations in gut microbiome may play a role in these AUDs. These patients exhibit gut microbiome alterations which seem to be aligned with severe craving. Craving for what? For alcohol. Heavy drinkers with dysbiosis can also have increased intestinal permeability. Here we go. This gut lining breakdown then leads to bacteria and toxins escaping into the bloodstream and entering the liver, where they can cause liver inflammation and organ damage. And if you perform fecal transplants from alcoholic patients to mice, it's been demonstrated that gut microbiome changes associated with alcohol were able to induce brain and behavioral disturbances, including impaired sociability in mice. So if you're taking from the feces transplant from alcoholic patients, human patients, you give that to normal mice, you begin to impair, amongst other things, the sociability in those mice. But to date, little was known about alcohol-related changes in the gut microbiome and cognitive and psychological correlates in early stages of alcohol misuse. So how do you look at those two things, microbiome changes and alcohol misuse? So examining effects of binge drinking, which by the way is the most common pattern of alcohol misuse during adolescence. Adolescents very much get into binge drinking. So by examining those effects, you could help identify early and novel biomarkers during this vulnerable period before addiction develops. If you can get them early, then you can avoid addiction. That's the key issue here. Get them before they enter into major adulthood with major problems of alcohol use. So in the present study, they looked at the microbiomes of 71 people, 18 to 25, who did not have the drinking issue. And the participants reported their alcohol use and if they had various neuropsychological problems. That was done through various evaluations. Craving was measured at baseline and 3 months later. And they took blood, saliva, hair samples to check for inflammatory issues, chylomicron and cortisol analysis, all of the things. And chylomicron, if you remember, is what you look at when you want to see the effect of things like cortisol on the brain part of the axis. Stool samples were provided by these individuals so you could get a metagenomic sequencing, and you could also look at their short chain fatty acids, which, as you know, is a real marker of what is going on in the gut. So the findings were the following. Those who reported more frequent binge drinking, that means in a 2-hour period, 4 or more drinks in women or 5 or more in men, they had microbiome changes correlated with greater alcohol craving. So that is adding to previous research that binge drinking was associated with greater blood markers of inflammation. Binge drinking is associated with distinct microbiome alterations and emotional recognition difficulties. So those two come together, alterations in the microbiome and emotional issues, which become a problem. Associations were found for several microbiome species with emotional processing and impulsivity. So like you'd see with a lot of studies, it's not all of the microbiome, but specific species that get affected. So craving showed a strong link with alterations in the microbiome composition and neuroactive potential over time. So what are the key takeaways? The most common pattern of alcohol misuse during adolescence is linked with gut microbiome alterations, even before an addiction develops. Binge drinking is associated with distinct microbiome alterations and emotional recognition difficulties. And there are several microbiome species that are associated with the emotional processing and impulsivity. So gut microbiome is important in regulating craving and social cognition. And a number of early biomarkers of craving have been identified. That's important. So the associations between emotional processing and the microbiome composition supports a growing literature on the gut microbiome as a regulator or one of the regulators of social cognition. And these findings are relevant for new gut-derived interventions specifically directed at improving early alcohol-related alterations during the vulnerability period of adolescence. And that's it.
What I said was it's a great study, and it emphasizes the connection between the gut and the brain. We've known for a long time they're intimately associated, and we're seeing here in this study that our clients with some alcohol problems might have it even worse in terms of the species of microbiota that affect mood disorders and emotional stability and things like that. Social cognition is the ability to get along in space with other people. That you can recognize facial expressions, for instance. That's all part of the cognition those are skills we'd have to get along well in society. So thank you again, Dr. Gonshor, for that. I'm sure some questions will come up. And I want to thank the rest of everyone for being here. I want you to subscribe, like, and share if you enjoyed this episode. Submit your questions, comments, and suggested topics, and grab a copy of the studies at fdntraining.com/studies. In a world propelled by science and technology, understanding the fundamental principles of science is no longer just an advantage. It's really a necessity, just what prompted us to put these together. So to thrive in tomorrow's world, become equipped not just with scientific knowledge, but with the ability to critically engage. And grab the scientific literacy course that FDN offers. You could find that at fdntraining.com/SM literacy. So join us again in 2 weeks on May 29th when we'll talk about persistent complement dysregulation with signs of thromboinflammation in active long COVID. And why, Dr. Gonshor?
Because science matters.